Hello, my name is Brian Tibble. I'm a senior majoring in molecular and cellular biology, and I'm in Dr. Stephen A. Boppart's biophotonics imaging lab working with his neurophotonics team. For this presentation, I'll be going over the calcium dynamics between cells with different retinal-based opsins. On this poster, we present the calcium dynamics of two common photosensitive proteins, channelrhodopsin 2 and melanopsin, and we transfect them into two mammalian cell lines, human embryonic kidney cells or HEK293 cells, and embryonic murine neuroectodermal stem cells or NE4C cells. We then monitored the cellular activity in response to optical excitation of these opsins using fluorescence microscopy of exogenous calcium indicators. Results display that channelrhodopsin transfected cells have different responses from melanopsin, namely by their amplitude and duration of photocurrents, which could be attributed to differences in their biochemical pathways. Optogenetics is a rapidly growing field utilizing different retinal-based opsins to enable cell-specific control of activity via light stimulation. In order to monitor the cellular activity and biochemical pathways in response to excitation of these light-sensitive transmembrane proteins, we studied the calcium dynamics of these opsins using video rate fluorescence microscopy of a green fluorescent calcium indicator, fluoro4. Calcium is a potent signaling molecule and one of the most important biological kinins that is critically important for a wide array of cellular functions. Opsins like rhodopsin or melanopsin are present intrinsically in mammals, specifically in the retina. Among these, melanopsin is found in intrinsically photosensitive retinal ganglion cells and has non-image forming functions such as its role in circadian rhythm regulation. In the figure on the left, you can see that melanopsin is a G protein coupled photopigment that promotes indirect activation of ion channels and has a slower, more prolonged response to light stimulation. In contrast, channelrhodopsin is an algae-derived ionotropic receptor that is gated by light. When these ion channels are activated via light, that channels open and allow ions to pass through, depolarizing the membrane. So we used HEK293 cells to compare the calcium dynamics of these opsins, and we also explored transfection of these opsins in differentiated NE4C cells. So first, we confirmed the expression of melanopsin and channelrhodopsin in HEK cells, and you can see that in the first figure in the results. We confirmed the transfection in HEK cells. We have GF green fluorescent protein that represents melanopsin expression, and yellow fluorescent protein that represents channelrhodopsin expression. And then we did the same thing in the NE4C cells, and we confirmed the expression through fluorescence microscopy for that as well. And we see yellow fluorescence protein confirming that the transfection was successful. Then we added calcium indicator, fluoro4, and retinoic acid to HEK cells, expressing these opsins, and we did functional calcium imaging using fluorescence microscopy. And you can see the functional calcium imaging of HEK293 cells with melanopsin over a 120-second period. We also plotted these calcium dynamics in both channelrhodopsin and melanopsin, and you can see the estimated delta F over F naught transients and the rise times in both opsins. And you could see that channelrhodopsin rise time was much more rapid than melanopsin. So we conclude that we successfully confirmed the expression of channelrhodopsin and melanopsin HEK293 cells and channelrhodopsin in NE4C cells. We estimated that the delta F over F naught transients and the rise times of calcium dynamics of these different opsins in response to light activation. We observed that channelrhodopsin had a weaker calcium dynamic response and a shorter rise times compared to melanopsin. These changes can be attributed to differences in their biochemical pathways because ionotropic receptors such as channelrhodopsin are expected to have smaller photocurrents and a more rapid activation compared to melanopsin, which is a G protein coupled receptor. For future work, we plan to continue calcium imaging experiments on HEK293 cells and begin calcium imaging on NE4C cells as well. We plan to explore how tailoring the order of different wavelengths of light interact with our sample affects its activation patterns. Prior work has shown the order does affect the output of channelrhodopsin expressing cells, a process known as coherent control. Seeing if this phenomenon extends to other opsins will be explored. We want to demonstrate that by altering the phase of our light sources for coherent control of these opsins, we can control the peak amplitude responses of single cells as well as their natural firing patterns. I would like to thank Carlos, Rishee, and Dr. Boppart for all the help throughout this project.